to understand a little bit about DNA and what the basis underlying what ATM does in the cell is all about. So I'm aiming to talk for about 30 to 40 minutes. So then we can have some time for questions. If you have burning questions as we're going through, it's far better that you understand it all. So do interrupt me and just ask a question. I want this to be as informal as possible. Okay, so just a minute, where's my slides? Oh, here we go. Okay, so what I'm going to try and do is get you to understand the terms DNA, RNA, protein and their relationship, because they're the essential things in the cell that's going to be relevant for understanding ATM. We'll talk about what the ATM protein does, but you'll learn to understand what I mean by the ATM protein. And we'll go through that a little bit more next time. And I'll talk about the types of mutations that cause AT, and then we'll move on next time. I won't be able to cover all of that today, some of the current treatment approaches and some of the research strategies. And the idea is for you to be able to get to the point where you feel you can understand some of these terms and get a feel for what the treatment approaches are and the research strategy is. Oh, wait a minute, how do I? Okay, so the way I've thought of starting to try and get you to understand the overall question is a human being arises from a single cell. That's a cell that's fused between the father's egg and the mother's sperm. And that cell can emerge and grow and divide and make a whole human being. And the human being has lots of different cell types. There's neurons, there's skin cells, muscle cells, all arising from that one single cell. So that single cell has all the information to make you into this whole human being, which is quite formidable when you think about it. So what I want to try and do, so the way that is done is the information is carried in a molecule. And what I want to do is try and explain that to you. I want to try and make this as easy. I know none of you are biologists, and to try and make it as easy as I can, I want you to think of, I'm going to do a little bit of analogy. Think of the UK as the organism that's made up of people who are the cells, and the people can be different. It can be workers, sometimes they're essential, sometimes they're specialized like fishermen. And that the written instructions, the information to make that whole human being is written down, and you can think of it as being like that written information is all there to be able to make all the workers, all the components that you need to make that whole human being. And the way this information gets carried to make the human being is in a code. It's in a code form, and that it's all there in that first cell, but it's actually in every single one of our cells. And the component, the molecule, so when I say molecule, I mean a sort of component that carries that code is called your DNA. You can think of Morse code, which is binary, dots and dashes, or you can think of an alphabet where there's 26 characters. And what we'll go through shortly is the number of characters that you have in that code. So the take-home message of this first bit is the DNA is the code carrier of the cell, having all the information to make a human being. And amazingly, it's only built up of 4 building blocks. These are ATCG, and I'm not going to tell you the names, you needn't worry about that. And we can think of them as colours: green, yellow, blue, red. And they're called bases. But they're arranged, and you can think of it like being the beads on a piece of string. They're arranged in a linear order, one-dimensional, and sometimes this this, if you like, a piece of string is called a strand. And in fact, and you have 6 by 10 to the 9, 6 billion of these building blocks. And that order is unique to any one individual. So the order of your 4 building blocks will be more similar to your children's than they will be to mine. 2 siblings will be more similar, but we're all different. That order is unique to every individual, and it will be the same in every individual cells, should be, that might change slightly, but the idea is that it's the same in every single cell of an individual. It's not quite as simple as this single strand here. And in fact, you have, can you all see that? Because I have your images come up slightly at the side. You actually have 2 strands that join together. And that makes it look a bit like a ladder, and your DNA is often called a ladder, and it has these 2 strands, so it's called a double helix. And so it's a double helix. It's got 2 strands. By a double helix, I mean 2 of those strands, and it's often just slightly twisted around, a bit like a spiral staircase, often called a helix. So that molecule, slightly twisted like that, is often looking like that in structure. And this is commonly how it's shown. So I've shown that point of it there. And let me just move this. Yeah. So the DNA, the code carrier, is a ladder-like structure just made up of those 4 building blocks. Sorry? Is that a question? No. Now, at the other end of the extreme, cells do tasks, and cells have worker molecules. They may be essential workers like nurses or fishermen, like nurses or doctors, or they may be specialized like fishermen's. And the worker molecules are called the proteins. And proteins are built up of amino acids, which you may well have heard about. You're supposed to get them in your diet. And there are 20, more or less, building blocks of these amino acids. So basically, a protein is made up of or a number of 20 building blocks. They're also arranged in a like beads on a string when they're built, but very rapidly there's these building blocks all interact with one another. There are forces behind them, and it takes on a 3-dimensional structure. So it very much folds into this 3-dimensional structure. The protein defective in AT is called AT mutated, or shortened to ATM. And it's a bit of an odd nomenclature, but what it means is that the protein that causes ATM, AT, the disorder, is mutated when it's causing that disorder. So it's called ATM even in a person that has normal protein. So a protein is a worker molecule, it's 3-dimensional, and it has 20 building blocks called amino acids. So I thought, just to try and show you what a protein looks like, I chose the coronavirus spike protein, because you may all have seen that or at least heard about it. Again, it's a worker molecule, and it's 3-dimensional, so you can see it twisted around, and the back may be different to the front, and the shape of it determines what function it might do, and it allows it to enter a human cell, and there'll be some part here that allows it to enter that has a shape that probably binds to something on the cell. So this is the ATM protein. It's very, very large. It's amongst the largest of proteins in the cell. The kind of diagram is different to here, and I couldn't find one that exactly resembled this, but it's built up of these many amino acids here, and it has the interactions will hold it into this form of shape. And we'll come back later to talk about exactly what that protein does. So the question then is, how do we go from that DNA code to making a protein? There's something called a protein factory that makes proteins, and it uses that code. But in fact, it can't use the DNA as a code directly. So it makes an intermediary code, which is called the messenger RNA or mRNA. There are several different forms of RNA, but this one is messenger. And you may well have heard of the messenger RNA in terms of the coronavirus vaccines, because some of them are using this to help create the foreign body that will make antibodies to. So the messenger RNA is a bit like the DNA, but the code is accessible to that protein factory. So just to go through this, your DNA is a long, long molecule. It has all the information to make all the proteins that we're ever going to make in our bodies, something like 10 billion building blocks. RNA is just a smaller amount of this, another code that is enough and has the information just to make one protein. So it's a subcode much smaller than the total DNA, and it's because and it can then be accessed by the protein factory. I'm getting some interference. I don't know if we all are. So RNA, slightly different to DNA, but it's also got 4 building blocks. The only thing is one of them is different. So they're slightly different, but I don't want to go into that. But you'll see there's a U instead of a T. It has one strand. So remember, I told you your DNA is double-stranded, but your RNA just has this one strand. And it's much shorter because it's only carrying the information to make one protein, unlike the DNA, which has all the information. And all of that makes it easier to be read by the factory. So the question you may be thinking about is, how do you go from a code that has got 4 building blocks to making a protein that has 20 possible building blocks? And the cell does that by reading that code in groups of 3. And it's called a triplet code. So, for instance, this is your RNA. You may have this sequence in your RNA, and this group of 3 will code for a certain amino acid. This will code for another amino acid, and so on. And if you do sums and you think of how can you arrange 4 building blocks in groups of 3, it comes out to 64 different possibilities. And I've said we've got 20 amino acids. So there is some redundancy. Some of these 2 different groups of 3 may code for for one protein. So there may be some copies of what they're coded for. This RNA molecule also at the beginning has a little triplet that says start reading me from here. And then it has one at the end that says stop reading me. So it knows when to start and when to stop. And then all that sequence in between will give you these amino acids that assemble into a protein. So your messenger RNA is a subcode, is extracted from the DNA, and it's the subcode to make a single protein. Now, the question is now, how do we go from DNA to RNA? And what you might think is that if that's a bit of your DNA sequence, your messenger RNA would simply be found in that sequence. Unfortunately, life's not as easy as that. And there are bits, so if here I've taken this sequence, which is a bit of your DNA, but I've highlighted some of it in capital letters. And it turns out that the cell uses bits to make that RNA molecule, and it cuts and splices. And you may have heard of splicing. And so it sort of takes a bit there, throws this away, takes a bit there, throws this away, takes another bit. So it's all there in your DNA, but there's bits of of what we call non-coding information separating these bits. And the cell somehow knows how to cut there and join it to there. And then that is read off in your groups of 3 to make your protein that all folds up in a 3-dimensional way. And I should say what I think is quite amazing is that the cell uses this same code. All species on this planet uses exactly that same code. It's universal in here. I'm going to show you the next slide, which named these bits that are used as exons and the bits that are thrown away as introns. You needn't remember that terminology, but I've put it there so that you can relate it to what I'm going to show you now. So this is really just repeating what I've said already. This is your DNA, and here you've got bits in dark blue that are going to make your messenger RNA. What I didn't tell you is that the first thing you do is actually make an exact copy of this DNA into RNA. And then you cut out these bits that you're going to throw away. And that makes your smaller RNA. Then you make your amino acid, which is like a chain, and it all folds together to give you the protein. The point I wanted to make here, which genes can have so a gene is the bit on the DNA that codes for a particular protein. And genes can have any number of exons and introns. There are a few that don't have any. ATM is a large protein, so it's a large gene, and it has 66 of these things where we're going to cut and splice, cut out this bit. So 66, what we call these exons, make up. So that's a lot of cutting and putting together. So I'm just going to go through, that's really the essence of what you've got to try and get from what I'm going to tell you today. But I'm going to go through a couple of things and add a bit more in, but we'll go through a bit of repetition just so that to be sure you've understood it. So your DNA, long linear code with all the information to make a human being, but it can't be read by the factory. It's arranged in a double helix, a ladder, and it's got these 4 building blocks. And it folds like that. Your messenger RNA is a subcode for making one protein. It's single-stranded, also has 4 building blocks, but slightly different from the DNA. And then your protein is the molecules that are going to do the job in the cell, and they fold very much and have these 20 building blocks. And remember, you go from there to there, you just make a fairly similar copy, but you cut and splice bits from there. And then in the protein, you go from this 4 building blocks to 20 building blocks by reading in groups of 3. Okay, so we've got DNA, it's used to make the messenger RNA. That's called transcription because you're just transcribing that information. And then that information is used to translate and make the protein. Now, proteins can't be made from another protein. They're made, you have to go back and use the information in the RNA or further back to get it from the DNA. You can't copy one protein from another protein. There's no machines that can do that because they're so tightly folded. So they're not important to the cell. They're dispensable. Some proteins will last quite a long time. Some others are discarded quite rapidly. A few proteins are made once, used and die. They're kind of kamikaze proteins. Your RNA, messenger RNA, can only be made from DNA. You can't make another molecule from itself. So again, it's not repaired. If it's damaged, throw it away. But the molecule that's critical is the DNA. It carries all that code and it must remain intact. And a lot of resources are put in to maintaining this molecule intact. And in fact, the cell is quite a dangerous place. It's constantly being, these molecules, all of them, are constantly being damaged by our own metabolism, byproducts from our own metabolism. So it's really important that the cell maintains the integrity of the DNA, particularly. And one of the things that ATM, or the major thing that ATM does, and the proteins encoded by AOA1 and AOA2, is to help maintain the integrity of your DNA. And that it's expressed in every single cell, because this is so important. Now, just to go back and give you a feel for numbers, proteins, we make about 20, we're able to make about 20,000 proteins. I think that's the estimate from these 20 building blocks, but no single cell makes that many proteins. As you become specialized, a liver may make certain proteins essential to the liver, a neuron may make some essential to the brain. I told you the messenger RNA has the information to make a single protein. So that's just a fairly simple molecule. The DNA, I've told you that it's long molecule, and that's not completely correct. It's actually divided into chromosomes, although they're long molecules, it's divided into chromosomes. And we have effectively, well, we have 22 direct pairs of chromosomes, all human beings, plus an X and a Y, or plus 2 X's. So effectively, we have 23 pairs of chromosomes, and they've got DNA in them, and this DNA is tightly bound, tightly woven in, sort of crumpled up, because you've got to get a lot of DNA. If you was to stretch out your DNA into the one molecule, it would be 2 meters in length, and it's all got to somehow fit into the cell. So it's very tightly wound up in these chromosomes. And as I said at the beginning, you've got something like 6 by 10 to the 9, 6 million of these building blocks. So I'll just go through this from the perspective of ATM. There's an ATM protein. I told you it's very large. It's made in every cell, and I kind of gave you a feel of what it looks like. The ATM messenger RNA is smaller than the total DNA, but it's still actually, by the standards of messenger RNA, is quite a large molecule, and it has about 8,000 of those building blocks. And I told you it involves 66 of those cut and splice events. And then for ATM, the ATM gene is on chromosome 11. It's a large gene because it's going to encode a large protein, but it's got large amounts of information that aren't used in it because you've got 66 of these cut and splice, and you've got all the information in between that that's thrown away. And we have 2 copies of chromosome 11. And as I said, it helps keep this whole DNA molecule intact. So now I thought I'd just, I know probably many of you have spoken with genetic counsellors and will probably have a feel for what a mutation is, but I thought I would just go through this from the context of DNA and what I've already told you. So a mutation is a change in the sequence of those building blocks. So for instance, here I've picked a T that may be changed to a C, a blue to a green, or whatever. I can't remember what colour I told you a T was. Now, what that may do in its simplest form is these are the bits that are going to, pieces of your DNA that are going to make your RNA. So instead of having a T there, you will have a C. And when you come to read off this little group of 3, you're going to put in the wrong amino acid. And that may be good, or, well, it won't be good, that's for sure. It may be very bad, or it may not be so bad. It could be that just one little change in your amino acid sequence doesn't affect things too much. Amino acids differ in charge in size, and some other qualities that affects how they fold together. So depending on what that change is, it may affect the folding of the protein. And it also may be in a very important part for the protein to function. We've heard talk about that with the coronavirus, where there are mutations that affect its ability to infect cells, how much it can hang on to the cells or get into the cells, or how it can be recognized by the antibodies, by the vaccines. So that will depend on how the particular nature of that particular change, how much it affects the structure of the protein, really. Some are not very severe, and so many of the 80 variants, which are milder, have changes that are of this nature. They're what we call missense mutations. So that's the first. So there are several different types of mutation changes that all result in a change in the sequence of your DNA, the sequence of those building blocks in your DNA. So I told you a missense mutation is this sort here, and it can be quite mild. Sometimes you can have a deletion of some of these building blocks. And what you may, if you think about that, what you may appreciate is that you can then put this reading, what we call out of frame. So instead of going CCC, UAG, which will be 2 and then more different amino acids, if you miss 2 of these, you'll go CUA and you'll be out of frame. You'll be putting in the wrong amino acid from that point onwards. And that's what's called a frame shift mutation. And that can be quite severe because you'll have completely different ones all the way down. The other kind of change is one of these cut and splicing events. Now, the question is, how does the cell know where it's got to put that together with that? And it's not easy. There isn't a single, you can't say, oh, the cell absolutely knows that. There are algorithms that can predict it, and pretty good at predicting it, but there aren't any defined rules for what you know when you go from there to there. Can be more geared by anything with some of the little sequences here and how they actually look to the machinery that does that cut and splicing. So sometimes, and for ATM, where there's quite a lot of those splicing events, 66 of them, 50% of mutations in 80 individuals are interfere with where you do this cut and splicing. It can, for instance, be that you just don't make your cut there, so you miss out this sequence here and go from there to there. That's one example. Another example is where you can create a splice site. So a mutation here makes something new and the cell thinks, oh, I'll go from there to there instead of from there to there. So there's various different types of these, what we call splicing mutations. The other important thing is that when you're reading your group of 3,
sorry, I'll cut off in a minute. Sorry, it's not urgent.
Perhaps whoever's got the phone going can just mute.
Sorry.
So going from this this group of 3 reading, it doesn't here actually it's gone pretty precisely, i.e. you start your new group of 3 there, but here you haven't done that. So the group of 3 can go across those what we call splice junctions. So if you've got a frame shift mutation, or sorry, if you've got a splicing mutation, that can cause the reading frame to be out of frame. So then you're putting completely wrong amino acids all the way down. So again, usually splice site mutations are quite severe. Okay, again, just kind of wanted to finish with this, which again, I'm sure many of you have been through with genetic counsellors who probably explained it much better than I'm going to, but I thought it might help you to see it in the context of what I've talked about. So I told you we have 22 pairs. We have pairs of all our chromosomes. That means we have 2 copies of all our genes. And ATM particularly is what we call recessive, which means just half the amount of protein is enough to be pretty healthy and normal. So an AT carrier has a good copy and a bad copy. And both the mother and the father will have a good copy and a bad copy. And when you make your egg or your sperm cells, you only land up with one of these copies. It's slightly different to the normal divisions in a cell, but when you're these type of divisions, you land up with just one. An egg has one, well, the egg has one chromosome, and it could be either one of the chromosomes, and the same for the sperm. So when those fuse, it's random. So you could have the bad copy and the bad copy giving you an affected individual, or a bad copy and a good copy, bad copy, etc., or 2 good copies making a normal individual. And that gives you the common, what we call 1:2:1 ratio, where of children, you should expect to have a ratio of 1 AT individual to 1 normal and 2 carriers. But remember, that is only something that holds when you take 1,000 individuals. For any one family, it may not work like that. And we know there are some families who've sadly had 2 affected individuals. Okay, so that's really all I wanted to cover here, just to go through it. AT is the disorder. ATM stands for AT mutated. It's the protein that doesn't function well in this disorder. Part of your DNA that codes to make the ATM is called the ATM gene. And first you make the ATM messenger RNA. That information is carried on chromosome 11, and it's called the ATM gene. So you've got your DNA, you're going to messenger RNA, and you're going to protein. So next time, what I'm going to try and do is tell you exactly what ATM is doing in the cell. I told you it helps maintain the stability of your DNA and how important that is, but we'll tell you a little bit more about exactly what it's doing. We'll talk about, may not cover this next time, but we'll talk about how its loss causes immunodeficiency and how its loss has the potential, provides the potential for cancer to arise.